I chose to tackle the problem of visual transduction because I wanted to find out how does the excitation of rhodopsin lead to a physiologic consequence. Now, at the time, we knew that there was signaling that went to the other cells of the retina and eventually to the optic nerve, but we did not really know what was the change in the retinal rod outer segment. But it seemed to me that this was a very attractive object of inquiry because here the outer segment of the rod cell, that's our dim light receptor, is packaged with the entire transduction machinery. So, if you take a retina and detached and just shake it like that, the outer segments fall off. And you can, in a day's work, you can harvest the machinery that carries out the transduction process. And so it occurred to me that this would be a wonderful way to get into the problem. And we then decided that the way we could enter the problem, the way that we might be able to make progress, is to look for a guanylyl nucleotide binding protein in rod outer segments. That is, the experiments of Liebman indicated that not only light was needed for the activation of the cyclic GMP phosphodiesterase, but GTP, guanosine triphosphate, was also needed. And so we decided to look for a GTP or GDP binding protein because we thought, such a protein could be the intermediary between photoexcited rhodopsin and the cyclic GMP phosphodiesterase. There had to be a link, and the requirement for GTP really gave us a clue of what the link might be. Now, at the very same time, people studying hormone action, people like Al Gilman, Rodbell, and others, were seeing that GTP was needed in hormone action, that the hormone itself was not enough to elicit the characteristic biological response, but that there had to be a GTP binding component, and that led to the birth of G proteins. So this was going on in the background, and it was certainly in the back of our minds. And so we decided to look for guanylyl nucleotide binding activity in the rod outer segment. And the experiment that Bernard Fung carried out was to incubate rod outer segments with radioactively labeled GTP. Now, the position of the label is important. It was the alpha phosphorus, the innermost phosphorus, that was labeled with P32. And so that would enable us to detect either GTP or GDP. Bernard then incubated the rod outer segments with the radio-labeled GTP and then washed away all of the GTP and then asked, was there any radioactivity left in the rod outer segments? And he found indeed there was. And what had happened was that the GTP that he put in, we now know, got converted to GDP, guanosine diphosphate, and that was what was permanently bound to the rod outer segments, permanent in the sense that Bernard was able to pull it down by centrifuging it. And then, in very short order, he carried out a really incisive series of experiments. First, he found that the entry of GTP into some binding site on the rod outer segment required light, that it wasn't enough just to have the GTP there. And it was that finding that enabled Bernard to make the very important quantitative study where he looked at the exchange process, and he showed that the kinetics of GTP entry coincided with GDP going out, strongly suggesting that there was an exchange process. And it was an exchange process that required light to trigger it. Bernard then went on and asked, well, how effective is light in leading to this exchange process? And then there was the wonderful moment where we were looking at the data and saw that it was just a tiny amount of light that was needed. We found that for a single photoexcited rhodopsin, there was an exchange of 500 GTPs for bound GDPs. This was the key finding. This was the first time that we saw that rhodopsin was doing something that led to a high degree of amplification in a very direct sense.